So, in chapter 7, we're going to talk about neoplasia, which literally means new growth. Typically we think about neoplasia as being cancer, but that's not always the case because not all growths are cancerous. Sometimes they can be benign. So, we start off on this slide talking about cancer incidence, not the incidence of neoplasia. But what we have, though, are two major sections here of estimated new cases and then estimated deaths. And what you'll find is that there's certain cancers that are becoming more common than others. However, those cancers like prostate cancer in men or breast cancer in women are still not the leading causes of cancer-related deaths in those individuals. So what we find then is that like prostate cancer and breast cancer are, highly treatable, especially if they're caught, early on. So when you look at cancer-related deaths, still in males and females, the leading cause of those deaths is lung and bronchus cancer. However, that's been on the decline for the last 30 years or so. Now, though, if you look at the second cause of death in males is prostate cancer and in females is breast cancer. So that's kind of interesting to note. We also find in both males and females, colorectal cancers are the third leading cause of cancer-related deaths, followed by pancreatic cancer. Now, what's kind of interesting about pancreatic cancer as being such a deadly cancer here, it accounts for 6% of cancer-related deaths in males and about 7 for females, is that if you look at the estimated new cases, it's one of the more rare cancers. So you find that the pancreatic cancer is near the end of this list in terms of its frequency of occurrence, but it's pretty high on the list in terms of its frequency of leading to death. So we find is that pancreatic cancer ends up being much more fatal as a type of cancer that you could, contract. Now, that's just important to note. Now, if you look down the list, the other cancer-related deaths include things like, liver and bile duct cancer, leukemia, esophageal cancer, and bladder cancer, lymphomas and kidney-related diseases as well. So what's interesting though here is we typically think about smoking-related cancers as only affecting the lung and bronchus, right? And that would kind of explain how it's so fatal here and also so common because there's still a lot of people who smoke. However, because when you smoke, those toxins are absorbed systemically, what you find then is that smoking-related cancers also include any of these other cancers. So when you start talking about like prostate cancer, colorectal cancer, esophageal, urinary bladder, kidney cancer, these also may be due to smoking-related toxicity. But you can't say for certain just based off this list, but there are some data that can suggest that. So when we talk about neoplasia, just in general, it just means new growth. So what we're saying here is that cells don't die off through apoptosis as they should. So what you find then is sort of a local area of accelerated growth where a tissue can increase in size and the number of cells. And what we find then is that if it's benign, these cells are pretty well differentiated. And what that means is they actually serve a lot of the same function as the original tissue, which you find these sort of dividing cells within. So an example of a benign growth could be something like adipose cells in a local area could grow very rapidly. And what you can get is something called a lipoma. Lipomas are essentially tumors or growths of fat cells. Now these fat cells look just like the surrounding cells. They're very well differentiated. They act like adipose cells, but they're different than the surrounding tissue because they have accelerated growth. And that's why we call it a neoplasia. And if it's benign, it's typically very well differentiated or resembles the surrounding tissue. Now, if it's malignant, it's less differentiated. And so one of the ways you can identify malignancies or cancers is that if these cells don't really resemble the surrounding tissue, and in fact, they might even have different functions than the surrounding tissue, then what we find then is that they're not maturing normally. They don't do the job that the tissue is supposed to do, and therefore can actually have some interesting sort of side functions that make it more likely to be malignant. So in terms of naming of tumors, remember, a tumor is just a new growth. So when you talk about benign tumors, those are going to end with oma. So when we ended with something like lipoma, right? So lipoma is a benign growth of adipocytes. Now, malignant tumors are what we call cancer. And so if it's a cancer of epithelial tissue, that ends in the name carcinoma, right? And so if you actually have a tumor or cancer rather of glandular tissue, that's going to end in adenocarcinoma. And if it's a tumor of mesenchymal tissue, which is any tissue that's derived from mesenchyme, which is all of your connective tissues, right? So that's bone, cartilage, blood, connective tissue proper, like the loose and the dense connective tissues, because they're all derived from mesenchyme. If they become cancerous, then those end in sarcoma. So just based on the name, you can kind of get a sense of what type of tissue is involved, and that will also tell you about the pathogenesis or the course of the disease as well. So if you ever hear the word carcinoma, right, and it doesn't have adeno in front of it, then you know that that's a tumor or cancer rather of epithelial origin. Adenocarcinomas are glandular tissue, sarcomas are connective tissue. So with the benign tumors, these are going to be very well differentiated. They contain cells that look like normal tissue cells. They may even perform the function of that particular tissue. And this could be a good thing or a bad thing. Sometimes if they maintain the normal function of that tissue, you can get sort of hyperfunction of that tissue. So an example could be something like a benign pituitary tumor could secrete hormones, and this oversecretion of hormones could lead to additional problems throughout the body. Now, benign tumors typically also grow slowly, and the reason why they grow slowly is they usually don't really recruit blood vessels that well, so that these cells aren't fed nutrients excessively, so they're sort of limited based on their nutrient supply. Benign tumors also typically are surrounded by a nice fibrous capsule, and this fibrous capsule, as the name suggests, encapsulates these cells within a local area, and it makes them less likely to spread or metastasize into the surrounding tissue. To be benign, they have to not infiltrate, invade, or metastasize or spread to the surrounding tissue. That's what makes them benign. However, people use the word benign to suggest that it's not dangerous, and that wouldn't be true. What we find is that benign tumors can damage nearby organs by compressing on them. They can, sort of put a lot of pressure on a particular organ or tissue, and that could cut off blood supply, cut off nerve supply, and ultimately lead to problems in the adjacent tissue. So benign tumors can be life-threatening based on where they're growing. Now, malignant tumors, on the other hand, have a lot of the opposite features of benign tumors. So if you remember, the benign tumors had cells that resembled the surrounding tissue, and they often carried on the same function. With the malignancies, these cells often don't look like the normal cells that you find in that tissue. They also don't normally form that particular function of that tissue either. And so what can make these malignant tumors or cancer even more vigorous is if they secrete signaling molecules, enzymes, or toxins that degrade the surrounding tissue, and therefore they're going to lack a capsule. And so they can also grow rapidly because they typically can recruit their own blood vessels into that tissue, so they can kind of feed themselves and grow more rapidly. In fact, what's interesting too is because they lack capsules, when the cells start to grow and invade the surrounding tissue, they typically have little legs of sort of the pathway of cellular growth. And the ancient Greeks recognized this as looking like a crab, and that's why cancer has the name cancer, because cancer means crab in Greek. And so it's based on the sort of the leg-like appearance of these growths. So malignancies will infiltrate, invade, and metastasize. Metastasize means that they spread to distant sites. So an example of metastasis would be like a colorectal cancer cell could spread to the liver because there's bloodstream that connects the two. And so metastasis is just spreading of these cancer cells throughout the body. Now, malignant tumors are also dangerous. However, they are more dangerous than benign tumors, mostly because they can metastasize and spread. They're going to grow more rapidly, and they also infiltrate the surrounding tissue. And so what we find then is that these malignancies or cancers can destroy surrounding tissues and even compress on tissues. And that's typically what leads to death, is not the cancer itself, but rather the damaging effects of this out of control growth on the organ systems that contain these cancer cells. So in terms of growth properties, what happens is that normally cells don't secrete a lot of growth factors if you're an adult. Now what happens though is that for cancer cells to really grow rapidly, they are typically going to express more growth factor receptors and or secrete growth factors in excess. Now, what this does is it accelerates the cell cycle so that these cells divide more rapidly. And in order for these cells to grow rapidly in a local area, they also need to lack cell density dependent contact inhibition. So if you guys remember going back to previous chapters, we talked about how cells can kind of modify their concentration within a tissue, because if the cells get too dense, they can initiate apoptosis of their neighbors. What happens in cancer cells, though, is that these cells can typically lack this density dependent contact inhibition, so that these tissues can get really dense with these rapidly dividing cells, and they don't initiate apoptosis of each other. Now, we also find that these cancers lack anchorage dependence, so they have anchorage independence. And what this means is that they have the ability to detach from their extracellular matrix and not undergo apoptosis. What happens, though, is that if a cell detaches from its extracellular matrix, it can undergo this anoikis, which is where a cell will undergo apoptosis if it's no longer attached to the place it needs to be. Now, with a cancer cell, in order for it to become malignant, it has to lack this anoikis, or at least have a little bit less of it. That way, when these cells do detach from the extracellular matrix, they don't undergo apoptosis, they survive and then spread throughout the body. Now, we can see too is that they don't have typically good cell-to-cell communication, and they're immortal. So if you remember going back to previous chapters, we talked about how one of the things that sort of determines the lifespan of a cell are telomeres. So remember, telomeres were the non-coding regions of the ends of your chromosomes that get a little shorter every time your cells divide. Now, what's interesting though is that cancer cells, because they divide rapidly, you'd think that they would run out of telomeres eventually and then essentially kill themselves. Now, what happens though with these cancer cells is they have active telomerase activity. So telomerase is the enzyme that actually makes these telomeres longer again. So they can regrow their telomeres, and effectively they can be immortal, right? In fact, there are some cell cancer lines that scientists use to study cancer that have been going on for 40 years. You know, they were able to get cancer cells out of people 40 years ago, and these cell lines have sort of carried on. They're immortal, and the reason being is these cells can regrow their telomeres, and they become longer, and therefore don't really end up sort of having that sort of age dependency that our other cells do. So with the cell cycle, what happens is that normally the number of cells produced should match the number of cells that die. Now with these neoplasms or new growths, that's not true. And so what happens then is you either see a decrease in apoptosis and an increase in the new growth of cells, such that there's acceleration of the number of cells in a local area of tissue. So we also should find that normally the total number of cells in the body should remain constant. And with cancer, that's also not true, because these cells are dividing more rapidly than they're perishing. So in cancer, growth fraction, which is the dividing versus resting cells, increases and the doubling time decreases. So what happens is that these cells can grow more quickly, and also they're less likely to undergo apoptosis, so that their doubling time decreases, which means that they can double faster, right? So if the doubling time is smaller, decreases, that means that they get to their doubling time faster. So like, let's say if it would normally take maybe blood cells to reach doubling time every 30 days or so, in certain types of blood cancers or blood-derived cancers, their doubling time could be on the order of days or weeks rather than months. So when we look at tumor growth here, it actually follows this exponential form of growth. So if we look at the number of doublings here over time, you might say that as these cells start to divide, and because they're not dying off, they're exponentially increasing in number. And so you'll see that even though it's exponential, because there's not a lot of cells initially, the tumor size isn't very large. But then we reach a point where, because there's a large number of cells and those cells are dividing, the doubling time really starts to have dramatic effects on the volume of the tumor, like the tumor size. And you see that there's really rapid changes that occur sort of later on in the progression of this growth. and such that you can see that from this point to this point, there's a dramatic increase in the size and number of this tumor, right? Or number of cells and the size of the tumor. If you compare that to previous growth cycles, it's a little slower. But because it's exponential, the bigger and bigger this gets, then the faster and faster it grows. And that's why it's important to catch cancer early on, because if you can catch it while it's still small, then it's less likely to have as many cells sort of accelerate to this in an exponential way, if that makes sense. So you're looking at basically growth of a tumor that's 0.5 centimeters in diameter into something that gets to 16 centimeters in diameter. So this is already several inches wide, if that makes sense. So growth factors we've talked about before with the cell cycle, because cells that have receptors for growth factors, those can actually kind of turn on or turn off their cell cycle. Now, we typically think about growth factors as being excitatory, such that they cause cells to divide more rapidly. So cells only divide when they're told to do so by these growth factors. Growth factors are usually going to be released as a paracrine molecule, so nearby cells can release growth factors that recruit the growth of other nearby cells. And this can cause stable cells to enter the cell cycle. So they're going to go from G naught back into the cell cycle and start undergoing cell replication and division. Now, metastasis is this idea that cells can break away from their original tissue and spread to a different site. So cells in a primary tumor can develop the ability to escape, travel, and survive in the bloodstream. Then they must exit the blood and then deposit into a distant site, which forms what we call a secondary tumor, aka metastasis. So if you were a cancer cell, think about which abilities would you need to survive in order to metastasize and proliferate, or just survive in the tumor, and which abilities would you need to metastasize. So to survive in the tumor, ultimately what cells need are nutrients, right? You can't have a living cell without nutrient supply. So if you were a cancer cell, you would need to have an adequate nutrient supply. And so what this means is that very vigorous cancers can recruit their own blood vessels, because what they can do is actually release growth factors like vascular endothelial growth factor that causes blood vessels to grow towards that cell. Now what happens is that cells that are hypoxic do release vascular endothelial growth factor, but cancer cells can also release a lot of this sort of excessively, and they can really get a lot of blood vessels to grow in the direction of those cancerous cells. And so this is going to help sort of ensure their survival, because as these cells divide and they start using more and more of the local nutrients, they also need to bring in their own nutrient supply. And so that would sort of increase their survival here. Now, in terms of metastasis, you would need a couple things. For one, you would need to have mechanisms to detach from the extracellular matrix. You would also need to prevent anoikis, which is the apoptosis that could occur after you detach from that extracellular matrix. And then you would also need to ensure that once detached, those cells can travel and metastasize, and then ensure their survival in that distant tissue. So there's a lot of factors at play here too. And then in both of these conditions, cancer cells need to evade the immune system. They need to hide from your immune system, because your immune system does a pretty good job of identifying cancer cells or tumor cells. And when that happens, your immune system will kill those cells. It causes them to undergo apoptosis. So successful cancers evade the immune system. That's actually how they're able to metastasize and spread. So when you look at metastatic carcinomas here, and if you remember, if it's a carcinoma, we're talking about how it's a tumor, or a cancer rather, of of connective tissue origin. So if you look here, we can see that this is a growth within a blood vessel, probably the aorta. And so if you look closely here, all of this is that growth of that tumor. And so what's happened is that connective tissue from another site metastasized, deposited in the blood vessel wall, and it started to grow within the wall of the aorta here. And so you can imagine then how that would start to compress on blood flow, and that can really sort of limit normal body function, because you're going to decrease blood flow downstream of this tumor. So this question here says, which gross anatomic feature of the lung tumor makes it more likely to be malignant? So, oh, sorry, that was actually the lung, not the aorta. But it looks like the aorta because it splits into two here. But I guess this is the trachea. So if you're talking about a lung tumor, what makes it more likely to be malignant? If the tumor grows quickly, if the tumor has poorly defined border, if the cells don't look like normal cells, or all the above. And what we know is that it's just B and C, right? So if we go back here, B says the tumor has a poorly defined border and the cells don't look like normal cells. So, malignancies can grow quickly, but that doesn't necessarily have to happen with malignancies. Not all malignancies do grow quickly. Some grow pretty slowly. Especially certain malignancies of the skin. They could be, sort of a malignancy that grows very slowly. It could be a mole that you think you've had forever, but it turns out it's actually a malignant melanoma. So the rationale here says gross anatomy can be viewed with the naked eye. Although rapid growth is a characteristic feature of malignant tumors, it can't be determined from the slide, right? We don't know if that was rapid growth or not. So the lack of clear border definitely tells us that it's cancer, and you can see that if you look back at the tissue here, because it's kind of hard to tell where the cancer stops and starts from looking at this picture. So when we talk about metastasis here, it's this idea that sort of cancer cells in a particular tissue can detach from their original mass, spread in the bloodstream, and then effectively leave the bloodstream and then reattach in some sort of distant site. Now, what's kind of fascinating, though, about metastasis is that not only do you need to have cells detached from their original mass, but you need to have intravasation, where these cells have to wiggle through the endothelium of a capillary and then enter the bloodstream. Now, what's interesting too is that if they can interact with platelets, they can form a traveling mass of cells. And what this does is actually stabilizes this growing sort of mass of cells. And these platelets are also sticky. So once these cancer cells have interacted with platelets, this sort of mass can stick to the wall of a distant site. And once that happens, you can have extravasation where these cells can leave the bloodstream and enter that new tissue. Once in the new tissue, they can actually attract their own blood vessels through the release of growth factors. And then because they're going to get their own nutrient supply, they're also going to grow way more quickly. So you might wonder, well, what are the genetic changes that can occur to make a normal cell become a cancer cell? And the idea here is that we have something called proto-oncogenes. A proto-oncogene codes for normal cell division proteins, right? Things like growth factor receptors, transcription factors, cell cycle proteins, and even apoptosis inhibitors. So these are normal genes. And normal cells use these proto-oncogenes in a normal way, right? So like, for instance, if tissues don't need to be growing very quickly, they don't release as many growth factors, and they don't need as many growth factor receptors. And they might only release cell cycle proteins when they're actually undergoing division if there's damage or replacement that needs to occur. We also have apoptosis inhibitors, where you can actually prevent apoptosis of normal healthy cells as well. Now, when proto-oncogenes mutate, then we call them oncogenes. So an oncogene is a proto-oncogene in its mutant form. So oncogenes are more likely to become cancerous, or actually cause the cell to become cancerous. So oncogenes come from proto-oncogenes, so they're already there inside of your genetic code. But the oncogene is the mutated form. So this could be where you might have like, tumor suppressor genes that are mutated and they no longer work. So you're more likely to get tumorous growths. And this can come from like insertions, deletions, or translocations. And these are types of genetic mutations we talked about in previous chapters. And so some examples of where proto-oncogenes become oncogenes would be like RAS or Philadelphia chromosome, or this HER2 neu. And so these are all examples of where normal genes in a cell, in their mutated form, actually promote the development of cancer. So we also have tumor suppressor genes, and what these do is they inhibit the cell cycle, they inhibit cell growth. Now, when tumor suppressor genes are mutated, that would actually, so if you're preventing the inhibition, then you're not preventing cells from dividing. So that cancer cells typically have lost, or basically have mutated tumor suppressor genes, so that their proofreading mechanisms aren't working, so they're more likely to get cancer that way. So this question here says true or false, oncogenes code for normal proteins used in cell division. And we know that's false, right? Because it's the proto-oncogenes that code for normal proteins. Oncogenes are in their mutated form, so that they're not normal, they're actually abnormal proteins. So oncogenesis is the development of cancer. And if we study this process of oncogenesis, it can start with a carcinogenic event. And so these carcinogenic events could be genetic mutations that can maybe due from chemicals, they could be due from radiation, they could be even caused by infectious microorganisms, like certain viruses can actually cause carcinogenesis. And they can also just be random too. However, too, sometimes cancer is heritable. So you might have already inherited a mutated gene, or allele rather, that can put you at a higher risk for cancer. And this is why you can have genetic tests for cancer. Like, you do genetic tests for like the BRCA1 and BRCA2 alleles, like with breast and ovarian cancer. You can know based on that, like what your risk of developing breast and ovarian cancer is. So what happens is we have this carcinogenic event that can convert a normal cell and have that can cause DNA damage of some sort, right? Now, what's interesting though is that normal cells can also help repair damaged DNA, and there are DNA repair mechanisms within our nucleus. So, if DNA damage does occur, it can be repaired and it can return back to its normal state. But let's imagine though there's a lot of this carcinogenic agent or event going on, such that you're overloading the cell's ability to repair its own DNA. And so if it sort of overloads that DNA repair mechanism, this DNA damage can actually progress to the next step, right? So if you fail to repair that DNA adequately, what happens then is if the mutation has occurred just in the right areas, we can get activation of growth promoting oncogenes, we get inactivation of tumor suppressing genes, and we can also get alterations in genes that control apoptosis. So what we're seeing here then is if these mutations occur just in the right genes, that can lead to unregulated cell growth. Now, this unregulated or differentiated cell growth can also lead to malignancy or neoplasm, right? And so that in that regard, we would call it cancer. So does this happen in a day? Not usually, right? This is going to be over the course of your lifetime through repeated exposure of carcinogenic events. It will slowly progress towards cancer. So the longer and longer you live, the more likely it becomes that you can develop cancer, just because you're having greater odds of being exposed to carcinogenic agents over the course of a lifetime. Now, you might wonder, well, what's the probability of this all happening? Like if the DNA damage occurs randomly, not always, but sometimes randomly, it has to be just in the right genes, right? Well, you have 100 trillion cells in your body. So with 100 trillion chances for things to go wrong, it's actually pretty likely that at least some of those cells are going to have mutations in these genes that could lead to cancer. So the lifetime risk of cancer in males is actually 1 in 2. And the lifetime risk of cancer in females is actually 1 in 3. So cancer is actually pretty likely in over the course of your lifetime. You're seeing 50% in males, 33% in females. So with growth regulation, we know it involves growth factors and growth factor receptors. And so what happens here is that with cancer cells, if they do secrete a lot of growth factors and they do express a lot of growth factor receptors, this can lead to changes within the genes of nearby cells or of the cancer cell itself. And this is done through this MAP kinase pathway. Now, the reason why we look at this particular pathway is that some of our cancer drugs target the MAP kinase pathway, because if you can turn off this MAP kinase cascade, then you can effectively prevent growth regulation based on growth factor receptors and growth factors, so that you can kind of slow down the growth of cancer and give your immune system a chance to try to kill off those cells. Now, if you don't, and this MAP kinase pathway works normally, then you're activating transcriptase, which is essentially turning on particular genes. So you're making mRNA for protein that could be like other growth factors. It could be more growth factor receptors. It could even be like cell cycle proteins that initiate these cells to start to undergo, additional rounds of cell division. So in terms of the stages of carcinogenesis, it occurs in 3 major phases. First of all, we need initiation, which is the moment that the initial mutation occurs in that time period where it's not repaired adequately, right? And so initiation could be caused by a lot of things. It could be random, first of all. It could also be due to, it could be from a carcinogenic agent or chemical. It could be from radiation. It could be from viruses. So there's a lot of things that can initiate mutations. Now, promotion is where those mutated cells are then stimulated to divide. And this could be stimulated by nearby cells that are like secreting growth factors. And when you initiate the division of already mutated cells, you're basically increasing the population of cells that are mutants, right? And so if at one point you only had one mutated cell, but then they're promoted to grow into 100, now you're 100 times more likely that some of those mutated cells may progress into more aggressive growths, right, through additional mutations. And so progression is where these tumor cells can compete with one another to develop more mutations, and they become more aggressive. And so it's almost like natural selection and evolution occurring in your body. If these cells are more aggressive and they grow more quickly, they're more likely to outcompete the less aggressive cells nearby for nutrients. And so there's sort of this battle of growth going on in your tissues once initiation and promotion have occurred. So if we look at the stages of carcinogenesis and we talk about normal cell lines, what can happen is these carcinogenic agents, whether they're chemicals, radiation, or viruses, can cause DNA damage. And once you have DNA damage and these cells divide, there might be some cells that, repair themselves once they do divide. But if they don't, then we talk about that as being mutated. And these mutated cells can therefore divide even further. And now you get a population of mutated cells, and we call that promotion, right? So they're promoted to divide here. Now what happens though is amongst this population of mutated cells, if there are some of these cells that can divide or grow more vigorously than others, that's what can lead to this progression where some of these cells can outcompete for the nutrients of other cells. In fact, you guys notice here too that these cells start to look different. And so it shows here that their nuclei are kind of oddly shaped versus the normal healthy cells where there's like a nice round nucleus and a very clear cytoplasm. So that's also true if you look at the histology of cancer cells. They even appear to be sort of abnormally shaped, and they have different numbers of chromosomes, and they even look mutated. So in terms of host and environmental factors for the development of cancer, for one, you can inherit it, and there's no way around that, right? So we do have some Mendelian inheritance of genes that can put you at a higher risk for cancer. So you start talking about things like BRCA1, BRCA2, as well as this RB gene for retinoblastoma. What we find is these are inherited from your parents. Now they don't determine that you're going to have cancer, but having these alleles or the forms of these genes puts you at a higher risk for those cancers, like breast and ovarian cancer or a retinal cancer called retinoblastoma. Now, it also is dependent on certain reproductive hormones. And what you find then is that if your reproductive hormones are like abnormally high, as with the case of taking things like birth control, or if you're like postmenopausal women taking hormone supplementation to support normal bone health, the presence of excessive reproductive hormones can also promote the progression of, cancers of reproductive tissues. And obesity also puts you at a higher risk for cancer. And it's kind of an interesting phenomenon here. You might think, why would obesity lead to cancer? Well, the idea behind obesity and cancer is that if you're obese, you probably have a high caloric intake. And if your caloric intake is high, that means you also have a high potential to feed cancer cells within the body. So if you're ingesting 6,000 calories a day, and there's a lot of calories left over. If there are some cancer cells in your body, and you're ingesting 6,000 calories a day, then you're basically feeding those cancer cells and potentially allowing them to divide even more quickly. So there's an interesting correlation between obesity and cancer rates, and they do positively correlate. So the more obese you are, the more likely you are to have cancer. Now, we also talk about how their immune system plays a role, right? So immune surveillance should normally help to kill off cancer cells because that's not wanted by the body, and your immune system can recognize that as being a potential threat. However, if you're immunosuppressed or if you have immunodeficiency that could be caused by drugs, radiation, viruses, or even heritable, what happens then is that you could be at high risk for cancer too. Even certain environmental factors like chemical carcinogen exposure, whether it's cigarettes, alcohol, or carcinogens in your diet, can also put you at a higher risk. Your lifetime exposure to certain amounts of radiation can put you at a higher risk, as well as certain viruses and bacteria. So the reason why viruses can lead to cancer is that they can either frequently irritate a tissue such that it progresses through this whole idea of metaplasia, dysplasia, cancer, or they can actually, if they're retroviruses, they can randomly insert their genome into the host cell genome, and that in itself can lead to mutations that promote cancer. And the same kind of goes for bacteria in terms of irritation. There are certain bacterial infections that can promote the development of certain types of cancers because of this frequent irritation, and because of frequent cell division from that irritation, you're more likely to have mutations that can result. So in terms of the local effects of tumor growth and how they're dangerous, we talked about how tumors can compress on adjacent structures. Now in hollow organs, they can actually decrease the size of the lumen. So an example would be like if you had a gastric tumor, that can start to compress on the lumen of that particular stomach, right? And so your stomach can't hold as much contents, and then you're going to see problems as a result, like malnutrition, nausea, vomiting, that kind of stuff. Now with blood vessels, what we can find too is you might get excessive bleeding or even hemorrhage if the blood vessel wall is compromised by the tumor that's compressing on those vessels. Now we can also see effusions that can result too. Remember, effusions are a leakage of fluid in the tissues, and that effusion could be due to an increase in pressure from the existence of that tumor in the particular tissue. So some of the systemic manifestations of cancer are things like anemia, and the reason why anemia is so common in cancer is that so anemia, by the way, is low oxygen carrying capacity in the blood. And the reason why you can have low oxygen carrying capacity in the blood from cancer is that a lot of cancers affect blood cells and bone marrow, where these red blood cells are made, and therefore you can see decreased number of red blood cells or mutated red blood cells such that they don't function normally. We can also see anorexia and cachexia also are common with cancer. Anorexia usually results from sort of the pain that can occur with cancer. It can result from the chemotherapy drugs that individuals can take for their cancer. And then cachexia is a wasting that kind of can come from anorexia, right? So if you're not eating as much, cachexia is sort of a wasting of your body tissues because the cancer cells are highly metabolically active, and they're going to use up your body's nutrients more quickly than your other cells. And so you see a wasting that occurs. And so individuals with cancer can actually get really thin, and they kind of waste away because their muscles get smaller, they lose adipose, and they become very, very thin. Now, with certain cancers, you can see fatigue and sleep disturbances. Again, this kind of co-occur with the cachexia or wasting. So you're fatigued because you don't have a lot of nutrients floating around because the cancer is eating them all up. And sleep disturbances can result if there's any kind of pain that's occurring or inflammation from that cancer. And then we can also see ectopic hormones that can be released too. So we'll come back to ectopic hormones when we talk about endocrine disorders, but we call these also paraneoplastic disorders. So if it's a paraneoplastic disorder, we're saying that they're cancer cells that are releasing hormones in a tissue that doesn't normally release that particular hormone. So an example would be like parathyroid secreting lung tumors. Some types of lung tumors can actually secrete parathyroid hormone, even though we know that normally comes from the parathyroid gland. So you can see odd things like hypercalcemia from lung tumors. So these paraneoplastic syndromes could produce hormone-like proteins like antidiuretic hormone or adrenocorticotropic hormone or parathyroid-related protein. And so these can result from cancer cells in tissues that don't normally make these particular hormones. And so these cancer cells can also produce proteins that affect clotting. So you can see abnormal clotting that can occur too. And there's also paraneoplastic neurological disorders where if tumors release signaling molecules into the bloodstream that end up in the brain, you can get neurological disorders like epilepsy and seizures from the existence of those cancers and the signaling molecules they release as a result. Now, in terms of cancer diagnosis and how we identify it, there's different ways we can look at this. For one, we can look at tumor markers. So tumor markers would be molecules that are either found inside those cells or secreted by those cells. And if they're elevated, like sort of out of normal proportion, that can give us an idea if they're tumorous. So one thing we can look for, an example here is prostate-specific antigen, or PSA. So this is a type of enzyme that's produced by the epithelial cells that line the ducts of your prostate. And there's normal amounts of this enzyme that are produced. But with prostate tumors or growth, they actually can hypersecrete this particular antigen. And so this is one of those markers you can look for to help determine prostate growth. Now we can also do cytologic studies where you actually take cells from a local area, like pap smear with the cervix, and you can do genotyping on those cells, you can do karyotyping on those cells, or even just look at the cells on the microscope and see if they look like normal epithelial cells from that location. We can also do tissue biopsy, like fine needle aspiration, where you might actually draw a sample of cells from within a tumorous growth to figure out whether it's cancerous or benign, and also figure out what type of cells those are, so your chemotherapy drug can be more targeted. You can also perform immunohistochemistry. So immunohistochemistry is where you have antibodies against certain proteins, and because cancer cells can secrete or create more of a certain protein type, you can perform immunohistochemistry to determine if there's excesses of particular proteins. Like for instance, estrogen receptors on ovarian cancer, you're going to find way more estrogen receptors than normal on ovarian cancer cells. You can also do microarray technology, and this is actually a really cool technique where you can measure sort of gene expression in cells very rapidly. And these gene chips can give you an idea of gene expression of, let's say, your tissue biopsy cells in question, and it can tell you about which genes are being expressed and or not expressed, and that can give you an idea of whether they're more likely to become cancerous or not. So some of the tumor markers we can also look for, to figure out like if they're cancerous or not, are things like oncofetal antigens or certain hormones. There's certain proteins and mucins. And so the oncofetal antigens we look for are things like alpha fetoprotein, or AFP. So this one's commonly used to help determine whether you have liver cancer or germ cell cancers. And so alpha fetoprotein is one of those types of serological markers you can look for in pregnant mothers to help determine if the baby's growing normally. Because this can tell you if the fetal yolk sac is actually dysfunctional or if GI structures are growing normally. If this protein's elevated, it's likely then that the fetus has some malformation of the development of tissues. You can also look for things like carcinoembryonic antigen, or CEA, and this is a marker of embryonic tissue of the gut, pancreas, and liver. And this can give you an idea of not just in fetuses but also in adults for colorectal cancers of the pancreas, lung, and stomach. And so you can look to see what their CEA levels are, and if they're elevated, it's more likely that they're at a higher risk for pancreatic, lung, or stomach cancer. Now, we can also look for hormones, right? Like for instance, you shouldn't find a lot of HCG, or human chorionic gonadotropin, in adults. That's a fetal type of hormone. Same with calcitonin. Like calcitonin levels should actually also be pretty low. Same with the catecholamines. So if you look at human chorionic gonadotropin, or HCG, we know it's a hormone normally produced by the placenta. However, after birth, you don't normally produce a lot of this particular hormone. And if you are, then that suggests it could be a tumor of something like the trophoblast, which can lead to further malformation of fetal structures, or even germ cell cancer of the testes. So in an adult, if they're secreting a lot of, adult male rather, a lot of HCG, it could be testicular cancer. Now remember, calcitonin doesn't really play a major physiological role in humans, but when calcitonin levels are elevated, this is actually a pretty good marker of thyroid cancer, because normally calcitonin levels should be fairly low. And then catecholamines, if you all remember, are epinephrine and norepinephrine, and these should be also within normal concentration, even if you're like sympathetically active, like your epinephrine levels don't rise above a certain concentration within your bloodstream. And so if you have a lot of these catecholamines being released, they actually can give you an idea of whether or not you have a tumor of the adrenal medulla, which is called a pheochromocytoma. So pheochromocytomas are diagnosed medically based on the blood levels of catecholamines. And then other specific proteins we can look at would be like monoclonal immunoglobulins or prostate-specific antigen. And so this can give us an idea of whether you have like something like multiple myeloma, which would be a tumor of the immune cells that make immunoglobulins. So if you're secreting a lot of immunoglobulins in excess, that could be multiple myeloma, which is actually a B cell tumor. And then prostate-specific antigen is an enzyme that's produced by the cells of your prostate, but this can also give you an idea of whether someone may or may not have prostate cancer. And there's other things you can look for too, like CA125 and CA19-9, and these can give you an idea of ovarian cancer or cancer of the pancreas. These are actually mucins, so they're actually molecules that are associated with mucus production. And so because there's mucus along these particular pathways, then you can get an idea of whether these tissues, like the ovary or the pancreas or the colon, if there's cancer there. And you can look at cluster or differentiation of those tissues as well. So for instance, CD antigens, which are present on leukocytes, can help determine if you have leukemia or lymphoma. So if you have a lot of CD antigens in the bloodstream, that could tell you if you have a certain type of leukemia or lymphoma, because that suggests that you have a lot of leukocytes, like excess number of leukocytes in the bloodstream there. So in terms of grading and staging, this is a way for clinicians to understand the severity of a tumor or cancer. And so grading is a microscopic examination of differentiation and the number of mitoses, right? So if you get a grade of 1, that means these cells are very well differentiated. And going back to the beginning of this chapter, we said that well differentiated cells are more likely to be benign. If you get a grade of 4, we say that these cells are poorly differentiated, which means that they don't really resemble the surrounding cells or tissue, and they might even have a different function. And so if they're poorly differentiated, that means they're more likely to be cancerous. And it's on a gradient of 1 to 4. So 1 being the best differentiated you can expect to see with any kind of new growth, and then poor meaning that they look very different than the surrounding tissue. And so 1 to 4 grade would tell you an idea about whether it's benign or potentially cancerous. So staging also kind of can come from clinical, radiographic, or surgical examination of the extent of spread of a particular tumor. And so this also helps determine the treatment and prognosis of that growth. So we have this TNM system. And so the TNM system is T for tumor size, and you can stage tumor size on a level of 1 through 4, 4 being a large tumor, 1 being small, N being the number of lymph nodes involved. If there's no lymph nodes involved, so that means you don't find any cancer cells in those lymph nodes, that's very less likely that this cancer is spread. And if you have an N of 3, which are 3 or more lymph nodes involved, then it is more likely that the cancer has spread. And then M is the number of identified metastases. M of 0 means no metastases identified, and M of 1 means at least one metastasis has been identified. And so TNM, which is this particular staging scale, can give us an idea of how we're going to treat this cancer and their particular prognosis, because there's actually data, like TNM data, from lots of people who've had cancer in the past. We also have an AJC scale as well, and it's a stage of 0 to 4, and this tells us about the size of primary lesion and the presence of nodal spread and metastases. But this TNM scale is the most widely used scale, especially here in the US. So in terms of cancer treatment, what we find is, there's surgical options, like they can be surgically removed. Radiation therapy is also a potential type of treatment. And the reason why radiation can be effective is that we have the technology and the ability to focus radiation at a particular level. So if you know exactly where these cells are, you can use focused radiation to kill those cells within a tissue. We also have chemotherapy, and these chemotherapeutic agents are usually cell cycle inhibitors, so they prevent the growth of rapidly dividing cells. We have hormone and anti-hormone therapy as well, which can slow the growth of these cells. There's biotherapy, which would be essentially using, like genetically modified viruses or genetically modified immune cells to target cancer cells. And then there's targeted therapy, which would be like a targeted delivery of drugs to a particular cell type. And so an example of a targeted therapy would be like packing drugs into a little sphere called a liposome or a micelle. And these liposomes or micelles can carry drugs that can either be lipophilic or hydrophilic. And what these can do is actually confuse with a target cancer cell as long as you have the right receptors in these little vesicles, and they can deliver these chemotherapeutic drugs or toxins straight to the cancer cells themselves. And this seems to be sort of the more effective technique. However, it's still more experimental. It's not widely used yet. But if we can figure out targeted therapies, then what you're saying then is you can just target the cancer cells and not affect any other cell of the body. And this seems to be the best option. But the technology is still advancing at this point. So in terms of chemotherapy's effectiveness, the whole goal with chemotherapy is to deliver chemotherapy in rounds, right? So you're not giving someone a chemotherapy or drug, constantly over a long period of time. What you're doing is you're giving someone a dose of drug, and it kills off a certain number of cells, and they might start to regrow again, but then you get another dose of drug, and it kills off another certain number of cells, and then they might start to regrow again. You get another dose of drug. And the reason why you want to dose their drugs, like in cell cycles, or sorry, it's not cell cycles, but just cycles of chemotherapy, is you need to give their bodies time to sort of recuperate from the drug itself. These chemotherapeutic drugs are pretty potent, and you couldn't just give someone a chemotherapeutic drug, like constantly over the course of weeks. So what you need then is to just sort of dose them over a time period, and that way you can kind of slowly kill off these cell populations and get them down towards, effectively zero. That's the goal, right? Is to kill them all off. But here's the problem, is that let's say if you stopped at the 4th dose and you said, "Oh, I'm done with this chemotherapy. I'm never going back again." But that you didn't kill off enough of those tumor cells such that you still had, approximately, 100 of these cells left. What that would mean then is if there were still a lot of these cells left, they could still proliferate and take off again. And so what you'd find is that maybe the tumor was almost gone, but if you missed that last dose of chemotherapy and you didn't get close to zero, these cells can take off again. And then you're back to clinically overt disease where there's actually signs and symptoms of the cancer, right? So this scenario here says Mrs. T has liver cancer. Her doctor did an initial molecular diagnosis and put her on targeted therapy, and the tumor shrank by almost 75%. 2 years later, the tumor was growing again, and this time it didn't respond to the drug. Last year, she was diagnosed with metastases in her femur, and that hasn't responded to the drug either. So Mrs. T wants to know why the cancer cells stop responding and if they're not the same cells, or are they not the same cells? And so I think what you could sort of tell Mrs. T here is that, if you initially had a particular type of cancer and it did respond to this particular chemotherapeutic agent, or targeted therapy rather. Now, the problem though is that her tumor only shrank by 75%, which means there was an entire 25% of cells of the original tumor that survived this targeted therapy. And so if these are the survivors, that means that they were resistant to this targeted therapy. And if they're resistant, then they're less likely to respond, once they start to regrow. And so that's kind of the key here with cancer treatment, is you usually don't want to have just one type of chemotherapeutic drug or one type of therapy, because you want to attack it from as many angles as possible. That way you don't have resistant cell populations that survive this round of drugs. Because if you do, then that cancer is going to come back and it's going to be even more vigorous and less sensitive to your drug. And it's kind of almost like antibiotic resistance for bacteria, right? Like if you don't take the full dose of antibiotics, you're more likely to have populations of microbes that are antibiotic resistant, and therefore if the microbes come back again and they regrow, they're less sensitive to that antibiotic. And so it's kind of true of these cancer cells as well. So with childhood cancer, it's usually going to affect hematological organs, like bone marrow, or nervous tissue, or soft tissues, or bone itself. And so we consider childhood cancer as sort of a clinical or critical period because the incidence is greatest during the first years of life. So there's really some things you want to watch out for in children. And that's because it occurs in the first years of life, and you really want to watch out for things like a chronic cough, right? Like if they're coughing really a lot, or if they have a sore throat that doesn't heal, that can tell you about whether they have a hematological dysfunction. If they have like fatigue, or if they have chronic infection, that can also suggest hematological dysfunction. One thing you want to look out for too is there's a type of cancer that's really common in children called retinoblastoma. It's heritable, and what happens is it's a cancer of the retina, and so the way you can identify it is that they get a change in the color of their pupil. So if you notice that a child's pupil starts changing color from dark to light, like white or red, it could be retinoblastoma, which is a nervous tissue growth, and you want to get that treated as soon as possible. So there's no early warning signs or screening tests, unfortunately, for a lot of these cancers. And so, really just want to observe as much as you can. And once you do observe these cancers, sometimes the damage is done. And so it's mostly just sort of damage control. But what is nice about children is they can typically recuperate from these, from this damage because they're still developing. Now, one problem too, though, is that childhood cancers can lead into adulthood problems, like late sequela. So, like for instance, a childhood leukemia can lead into adulthood, autoimmune disease, or it can lead into adulthood anemia, or other problems there too as well. So, and sometimes the treatments for the childhood cancer that in itself can lead to further problems. Like if you give children, chemotherapeutic drugs, because of the fact that cells can become resistant to that chemotherapeutic drug, it's going to make it more difficult to treat cancer again if it shows up in those adults that had childhood cancer. So that's the idea here of having late sequela.